Hello, everyone. Hello, welcome. This is the first episode of a project that I like to call STILTS. It's science taught in little tiny segments. So essentially the idea of this is I'm going to sort of give you a brief overview of some sort of scientific topic. And for this one in particular, it's going to be how proteins are made. So essentially I will just talk about it's not going to be overly detailed. I want everyone to be able to understand what's going on. Anyone of every education level. I am going to give a caveat with this. These presentations are not made to be used for studying your tests. There's much more details you need to know for your tests. This is only to generally inform you. My name is Caprolux, and I welcome you to learn with me on this scientific journey. So let's get started. How proteins are made. Here's a simple brief overview. Our DNA is where we get all our information. It's how every protein is made. And then that information is transcribed into RNA. So essentially it's just the process of the information goes from DNA to RNA and then to proteins. So this is how proteins are made, essentially, or at least where they get the information from. I will be omitting a few things, but hopefully this will give you the idea. So to start out with, we need to know how the DNA is structured. So our DNA is structured with 4 what we call nucleic acids or bases. And that's thymine, adenine, cytosine, and guanine. TACG. Just 4 simple letters. And that contains all the information for us and how we're made and how we're created. So you'll see here that T and A are always paired off with each other. And same for C and G. So there's always a pair. Whenever you have a T in your DNA, since it's made of a double helix, it's always paired with another base. So T to A to T, C to G, G to C, and so on. You get the idea. But then you may ask yourself, well, how do we get all this information from only four simple letters? Well, the overview I showed you earlier of DNA to RNA to proteins, there's 2 processes. For DNA to RNA, this is called transcription. And so transcription is when the RNA is being built based off of the bases in the DNA. And translation, which I'll go over later, is the process of that RNA that was built from the DNA being made into, you take the amino acids and create a polypeptide. So let's get into transcription. So here we've got our DNA. We have 2 strands. This is your chromosome that your DNA forms. So it gets unraveled, and then there are 2 strands. There's a template strand and a non-template strand. The template strand actually reflects the non-template strand for the actual bases that are being used. So you'll see here, this RNA polymerase takes the template strand, and it then starts pairing everything together. So C to G, T to A, G to C, and this right here. So RNA actually doesn't use thymine. It uses a slightly modified base called uracil. But for the simplicity of things, you can just imagine a U as a T, but for RNA. So C to G, T to A, G to C, and A to U. So we'll continue along this on whatever gene it's trying to transcribe. So then we'll get this long stretch of RNA coming from the DNA. So just like this, here's the non-template strand, here's the template strand. And so you'll see the RNA that's created from the template strand actually matches the non-template strand. See? AA, TU, which again, U is just T but for RNA. GG, AA, TU, CC. So the RNA is actually building the non-template strand, but you don't need to worry about that so much. So we've got our transcribed RNA, and we've got our series of bases that we want to use. So now, how does that get transformed into a protein? So it's a process called translation. Just so you know, there's actually multiple kinds of RNA. So the RNA that we see here is actually mRNA. mRNA is what is taken from the DNA. Now, these big blocks here, this is actually RNA as well. But this is tRNA. And with the structure of tRNA, there's always 3 bases. There's 3 bases which are called codons. And this is how the RNA is actually read. 3 bases at a time. So you see AUG, that's 1 codon. Same for UGG, that's another codon. And so on and so forth. So the mRNA is brought to ribosomes, which help create the protein. And what happens is the mRNA is pushed in, and it's read from 5' to 3' end, which is just the ends of nucleic structures. And so we start with, also each tRNA is attached to an amino acid. Each codon has a specific amino acid that it belongs to. So the tRNA with the amino acid is brought in, and it pairs off with this. So as you can see here, with this tRNA, AUG would then be UAC. And then with that tRNA, it's paired with methionine. So the tRNA brings in the amino acids, and then right here in the middle part of the ribosome is where the amino acid is attached to the next amino acid to start building the protein, or the polypeptide as it's called. So this will continue on almost like a printing machine, where the RNA just goes through, tRNA binds to it, and then an amino acid is brought and added onto the polypeptide. So then we get essentially a string of these amino acids. And to briefly talk about codons again, so like I said, each codon is specific to an amino acid. Also, as I said earlier, there's only 4 bases, but the fact that they're read 3 bases at a time actually can create all these combinations of codons. So that actually gives us 64 combinations for different types of codons. This is a codon table, which makes it very easy to read what codon you're going to have, or what amino acid. So you just take your first letter, take your second letter, and then your third letter. So U, and then U. So that'd be this amino acid. So each of these would be with, its respective amino acid. There are a couple of exceptions, or not exceptions, but specific ones. So AUG, methionine, this is actually at the beginning of every single protein. This is at the beginning of every single gene, and this codon essentially codes for the start sequence for what needs to be transcribed in the DNA. And then also, there are actually 3 stop codons. If we were to transcribe all of our DNA, then that would go on forever. There's billions of bases in the DNA. So each gene also has a stop codon with it. And then once this codon is read with the RNA, then it'll stop translation. It'll stop transcribing. And so, to go back, we had our DNA. We made our RNA from our DNA, got that transcribed, and then we created this polypeptide. So now we have all these amino acids in a chain. Now, the interesting thing is how different proteins are made. For example, we have these things called exons and introns. Exons are what gets transcribed. Introns are sort of what we call junk DNA, quote unquote. In this example gene, there are 5 exons. So they're transcribed, and the RNA has these 5 exons, and also all the introns in between. Now these introns are actually going to get cut out. So we're just going to have the colorful blocks you see here. But there's this very interesting thing called alternative splicing, to where we combine different colored blocks to be able to make different proteins. So say for example, this gene has 5 exons. Well, we can make all kinds of proteins with that, because we could combine 1, 3, 5 and make this protein. We could combine 2, 3, 4 and make this protein. Or 1, 2, 5 and this protein. So that just creates even more possibilities with the amount of proteins that can be made in your body. So we've gone from our DNA to our RNA to our protein. But they actually don't automatically go like this. See, there's a structure to proteins. So here's the amino acids that we built. There are actually 4 levels of structures for proteins. There's primary structure, secondary structure, tertiary structure, and quaternary structure. 1, 2, 3, 4. And so we have our polypeptide, but it actually has to be folded in a specific way to be able to create this protein. So see here, this is one example of a secondary structure. It creates a helix. So it makes a bunch of helixes, or whatever other secondary structures, and then this creates the tertiary structure, where it's all folded in on itself. And the quaternary structure, which itself will be the protein, is actually the aggregation of 2 or more polypeptides. Now there are examples of some proteins being only 1 polypeptide and such, but for the majority of proteins, there are multiple polypeptides. Everything in a protein has to be folded exactly the right way, and that's usually done with different chemical bonds to be able to create this quaternary structure. But to review everything, we have our DNA, then we go to our RNA, then we create proteins. The DNA is transcribed into RNA, and then the RNA is translated into the polypeptide. And then that polypeptide needs to be folded and then combined with other polypeptides so that it creates a protein. And that's it. That's the general summary. Of course, there's a lot more details that can go into that. So if you're interested in that, I grabbed this additional reading and info, some reading and some videos that you can watch if you're curious in it, that I'll put in the description of this video so that you can go check those out. But yeah, if you have any questions, you're welcome to come ask them live when I'm giving this presentation at twitch.tv/Caprolux, or you can leave in the comments of the video, and that way I can answer questions on both platforms. That's it for this episode of Stilts. Hope it was very informative to you, and I hope you are able to use your stilts to elevate your knowledge to a further level. And that's it. Goodbye, everyone.